Hello students. So today we will discuss about the topic translation. Okay, and already we discussed about transcription, and now it is the turn of translation. What is translation? Okay, formation of a polypeptide chain with the help of the information brought by messenger RNA from the DNA. So polypeptide chain means what? A protein will have different types of amino acids, and the sequence will be different, and how they have to be arranged, what is the position of a particular amino acid, everything is present on that messenger RNA, and it has brought this information in coded language, and we have come to know that this coded language is in the form of triplet codons. Okay, and already we discussed about the genetic code and some properties of genetic code, and the initiation codon is AUG, and it codes for methionine in in eukaryotes, methionine, whereas it is formylated methionine in prokaryotes. Okay, that is only the difference. Okay, formyl group is present with methionine in prokaryotes and is absent in eukaryotes, but the codon is same for both of them. And remember that this codons are present contiguously over this, and you can see that this is the mRNA, this is the 5' end, and this is the 3' end. Okay, and in the beginning of this mRNA, you will find the initiation codon. Okay, and towards this 3' end, you will find a termination codon or nonsense codon, and we already discussed that UAA, UAG, UGA are the terminating codons. And remember, any one will be present at towards the 3' end. Okay, and now let us see how this translation process will occur. There are different steps, and we can say there are three steps in this translation. One is, first of all, amino acids should be activated, so that's why activation of amino acids is the first step. And the second step is charging of tRNA. Means what? These amino acids which are activated should be attached to the tRNA. So that is what charging of tRNA. And then the third step is formation of polypeptide chain. And in the third step, again, you will find three substeps. One is chain initiation, and the second one is chain elongation, and the third one is chain termination. Okay, and actually, this is a very complicated process. And first, let us say the activation of amino acids. So, in the cytoplasm, many amino acids will be there, and we can call it as what? Amino acid pool. And all these amino acids, 20 different types of amino acids are participating in protein synthesis. All of them should be become active so that they can get attached to a specific tRNA. Okay, so that's why, for example sake, I have taken one amino acid, and I denoted this amino acid as what? AA1. That means what? A amino acid 1. Amino acid 2, amino acid 3, amino acid 4, amino acid 20. So like that you can write here. That means this step is common for all amino acids. Okay, and it requires, for activation, energy is required, and every amino acid will make use of for the energy in the form of ATP. And remember that in competitive examinations, they will ask you how many ATPs might have consumed for the formation of a polypeptide chain composed of 100 amino acids. Very simple. You see, each amino acid requires what? 1 ATP. So 100 amino acids are there. So how many ATPs have con- got consumed? 100. Like that you remember. So ATP. And there is one enzyme is required. That is what? Amino acyl RNA synthetase enzyme. Amino acyl RNA synthetase enzyme. And I wrote E1. That is what? Enzyme 1. Means what? Even these enzymes are specific. For every amino acid, one specific mRNA will be there. So then, how many types of amino acyl tRNA synthetase enzymes will be there? 20 types. Because 20 types of amino acids are there. So 20 types of enzymes should be present. Okay. And it is activated by magnesium. That's why I wrote here enzyme 1 plus magnesium. Okay. Now, as a result of which, with the help of this, one complex is formed. That is what amino acid 1, enzyme 1, and two phosphates have been provided energy, so they are released. So that's why this is the complex AA1, enzyme 1, AMP complex, which is called amino acyl adenylate complex. Amino acyl adenylate complex. Okay, now that amino acyl adenylate complex, in that the amino acid is already activated. It is with energy, and now it gets attached to one particular tRNA, that is stands for RNA. You can see here. I brought this one down here, AA1, E1, AMP. That is what amino acyl adenylate complex. Plus tRNA 1 I wrote. Means what? Even tRNAs are specific for amino acids. So for amino acid 1, tRNA 1. For amino acid 2, tRNA 2. Then for amino acid 3, tRNA 3. Like that. Okay. And now it gets attached, and already in the previous video we discussed about the structure of the tRNA. It has 5 prime end and 3 prime end, and the 3 prime end is also called as CCA end, and to that CCA end the amino acid will be attached with the help of what bond? Ester bond. And this bond formation is called what? Esterification. Okay, so you can see that amino acid 1 is now attached to tRNA. So one complex is formed here. That is what? Amino acid tRNA. Or you can call this tRNA as what? Charged tRNA. Charged tRNA. Or you can call this also as what? Amino acyl tRNA. Amino acyl tRNA. And even these amino acid tRNAs can be called by these amino acids which are connected to that particular tRNA. Suppose tRNA with methionine, met tRNA, tRNA with glycine, gly tRNA, leucine, leu tRNA, serine, ser tRNA. So valine, so val tRNA. I hope you're understanding. So, based on the type of amino acid, you can recognize how many types of tRNAs. 20 types of tRNAs. Okay, now you see this reaction is over and the charged tRNAs are formed, and now what will be released? That AMP, which is here, AMP is released, and enzyme also becomes free. So now, once all these tRNAs, now they're all charged and they're all ready for the next step. And the next step is what? This formation of polypeptide chain. And for this, they have to reach one factory. And what is that factory? Protein factory. So, what is that protein factory of the cell? Or cellular factory of proteins is ribosome. And you know that in prokaryotes, the ribosome is of 70s type, and in eukaryotes, it is 80s type. And the ribosome will have two subunits, and the subunits, when the ribosome is in inactive state, they are present in dissociate form. And you know that the smaller subunit is 30 years, and the largest subunit is 50 years. That is what, both of them together when active form, so it becomes what, 70 years. This you will find in prokaryotes and also in the chloroplast and mitochondria. And let us say, in the case of eukaryotes, the smaller subunit is 40 years and the largest subunit is 60 years. And both of them, when they are active, when they are together, they make 80 years type of ribosome that is present only in eukaryotes. Okay, and this ribosome will provide two sites. One is what, P site or peptidyl site, and the other one is what, A site, that is what, amino acyl site. Okay, just remember P for stands for what, peptidyl site, and A is for amino acyl site. Okay, now let us say, actually I do the diagram completely here, but in the first step, sub-step, chain initiation, what will happen? This mRNA having that information now first binds to the smaller subunit of the ribosome. And actually this won't be there, and immediately the first codon, but of course you know that the first codon is AUG, and that codes for what, methionine. So which will come and attach to this the met tRNA. I wrote, of course, AA1, and I have denoted these things, XXX for the first codon, YY second codon, and ZZZ for third codon. Okay, like that. And you see, first this met tRNA will bind to this first codon. Okay, and you can see it is carrying what, this first amino acid, and immediately this largest subunit, that is largest subunit of ribosome, will attach. Now a complex is formed. This complex is called initiation complex. So now, can you tell me what are the things that are present in the initiation complex? Okay, mRNA, smaller subunit of ribosome, and larger subunit of ribosome, and this initiation tRNA means what? Methionine tRNA. And you can see that it has occupied which site? P site, that is peptidyl site. And in the peptidyl site, of course, so you will find one enzyme called what, peptidyl transferase, and that helps in the creation of what, peptide bond. Now let us say that is what, chain initiation, and this also requires what, initiation factors. And you just remember, 3 initiation factors are involved in the prokaryotes, whereas 9 initiation factors are involved in eukaryotes. Okay, and now let us say this is the first one. And I hope I did not tell you this smaller subunit is 30 years in prokaryotes, and it is 40 years in eukaryotes. And the largest subunit is 50 years in prokaryotes, and the largest subunit is 60 years in eukaryotes. That you have to understand. Okay, now let us say what will happen. And now the second, this is P site, and this is A site. Okay, so let us see how this elongation will start. Okay, so now this A site is exposed to the second codon. So let me say this is first codon, and this is second codon, and this is third codon. Okay, so now A site is exposed to this second codon, and according to that codon, that particular tRNA will bring that amino acid. Just for understanding, I wrote AA2. Okay, and now both of them are here. So you can see that AA amino acid 1 is in the P site, and amino acid 2 is in the A site. And I told you already, here there is one okay, enzyme is there, that is what, peptidyl transferase, which is nothing but rRNA. So don't forget that ribosomal RNA will show catalytic action. So it is 23S rRNA in prokaryotes, and whereas it is 28S rRNA in eukaryotes will behave as peptidyl transferase. Okay, now you can see that both of them. So now the peptidyl transferase will bind them, and this bond is formed between the carboxyl group of first amino acid and the amino group of second amino acid. So the carboxyl group of first amino acid and amino group of second amino acid, and the bond is converted peptide bond. So now what will happen? These bonds will break, and as a result of which, what is released, this tRNA will come out. Okay, now you can see that what will happen, the ribosome will now move towards what 3 prime end. So the ribosome is moving from in what direction, 5 prime to 3 prime direction. And it won't move very fast. That means what? It moves in a stepwise manner or codon by codon. So earlier it has occupied two codons. Now you can see that it is okay, moving, skipping first codon, and now it is occupying the third codon. So now you can see that the first there is a first and second codon, and now you can see now you will find what is second and third codons here. And now, because of this movement, you can see the position of AA2. Earlier, AA2 was in A site, but now it has come to the P site. Okay, you can see there amino acid 2 was in A site. Now, because of the movement of ribosome, it has shifted to what P site, and this is called translocation. This is called translocation, and now you see, you can see the amino acid 2 is in P site and amino acid 3. So now actually amino acid A site is exposed to third codon, and because of which according to that codon, okay, another tRNA will come, that is okay, tRNA3. Okay, and here again what I formed, okay, it binds to this codon, okay, with the help of the anticodon. And now you can see that second amino acid is in P site and third amino acid is in the A site, and the peptidyl transferase now again will do the same thing, that it binds these two with peptide bond. So now the bond is formed between the carboxyl group of amino acid 2 and amino group of amino acid 3. So like that, and this one, this tRNA which is in the A site, tRNA which is in the P site now will come out. Now again the ribosome will move forward, and then when like that, what will happen? The third amino acid and now fourth amino acid will come and join. Fifth amino acid will come, and sixth amino acid, seventh amino acid, eighth amino acid, ninth amino acid. Whatever the number of sense codons are there, the same number of amino acids will be connected, and a long chain is formed. Now let us imagine that this is the 10th codon here. This is the 10th codon, and let us assume that this is the last codon on mRNA, last sense codon. So already amino acid 10 has come here, and it is connected to the ninth amino acid. And now this one is expecting one more amino acid, but the thing is that now it has come to this codon, and this codon is okay, terminating codon, and it doesn't have any tRNAs. That's why, as no tRNA is there, so now some okay, uh, release factors will occupy this site, and so that and remember that release factor ERF1 in EK, that means eukaryotes, ERF1 in eukaryotes and RF1 and RF2 in prokaryotes will are the release factors. So in eukaryotes only one release factor is there, but in prokaryotes two release factors will come, and whenever they come, you see, so this tRNA will get disconnected and the tRNA will go off and the whole chain will come out, and these smaller subunit and the larger subunit of ribosome also will get separated. In that way, so what is formed, a chain is formed, and this is called chain termination. I hope you are understanding these steps. First, chain initiation, and then chain elongation, and then chain termination, and finally we saw about a formation of a polypeptide chain. And remember, some questions will come in the examination. Suppose the mRNA is with 100 codons. mRNA is with 100 codons, and after translation, a polypeptide chain will be formed, and that polypeptide chain will have how many amino acids? So just keep in mind that if there are 100 codons, so out of 100, 99 are sense codons, and there should be one termination codon. So that's why there is no amino acid for termination codon. That's why when the translation is completed, so the polypeptide chain will be composed of how many amino acids? 99 amino acids. Okay, and like that they will ask some more questions like this. So suppose mRNA is with 100 codons, and it is made up of how many nucleotides? And don't forget that codon is triplet codon. So each triplet codon is with three nucleotides. So just simply you multiply it with 3, 100 into 3, 300 nucleotides. Okay, so like that, if you understand the topic, so it is very easy to solve such simple problems that are asked in the examination. Okay, right. So that is about the translation. Translation.